Brainiac. Today we're gonna see a biological video. And today we're going to talk about the kingdom system of classification. As we know, many scientists worked in the biological fields and put forward many forms of classifications. Those famous classification theories are as follows. The first one would be the Aristotle classification, and the second one is the Linnaeus classification, also known as the two-kingdom classification. The third one is known as the Heckel classification, also known as the three-kingdom classification. The fourth one is Copeland classification, also known as the fourth kingdom classification. The next one is the most important one, the Whittaker classification. We will study about this deeper. This classification is also known as a five-kingdom classification. And the final one is also important, the Woose classification, also known as the six-kingdom classification. We also honor Woose for another thing of discovering the three-domain classification. We will talk about that later in this video. If you take a deeper look at Aristotle's classification, we can clearly see that mostly morphological features are considered, and the next considered fact is the basis of RBC. That point comes under the kingdom of animals. Aristotle has clearly observed the difference of morphological features between plants and animals, but mostly morphological features come under plants in this classification. Considering plants' morphological features, Aristotle divided plants into three, as herbs, shrubs, and trees. The main form of morphology considered in this plant classification is their height. Herbs are plants with the smallest height, and shrubs are plants with an average height, and trees are plants which are the tallest. And if you consider animals are considered in the basis of the presence of red blood cells, also known as RBC. Aristotle divided the kingdom Animalia into two, known as Anima and Inanima. Anima is the group of animals in which RBC is not present. But if you consider the group Inanima, in this RBC is present. And the next form of classification we should study is the Linnaeus classification, also known as the two-kingdom classification. He also did the same thing what Aristotle did. He also divided the environment into plants and animals, but not as Aristotle. He did a significant change in this classification. He identified some significant points which can diversely show these two kingdoms. If you look at this table, you can clearly observe what types of differences did Linnaeus observe. The first difference is the cell wall. If you consider plants, there is a cell wall present, and it's made of cellulose. But in animals, they do not have a cell wall. And as for plants, they do not locomote, but animals, they do locomote. The next difference is nutrition. Plants are autotrophic. Autotrophic in the sense holophytic. It means they are photosynthetic and they can produce their own food. While animals are heterotrophic, they depend on another animal or another organism for food. And the next difference is the response to stimuli. Plants respond to the stimuli slowly, but animals respond to the stimuli faster. But in this case, on the response of stimuli, there is one plant that shows fast stimuli responses. That plant is known as mimosa. A mimosa plant would look like this. For clear understanding, you also can call mimosa as touch me not. If someone go and touch a mimosa plant, it automatically closes its leaves. The next difference is the contractile system. Plants do not have a contractile system while animals do have. If you consider examples for plants and animals, examples would be bacteria, fungi, algae and higher plants in the plantae kingdom, while protozoa, invertebrates and vertebrates in the Animalia kingdom. There are several drawbacks in this classification. As you can remember, bacteria was in the kingdom Plantae. Along with it, there were fungi, algae, and also higher plants. If you clearly see, bacteria is prokaryotic, while fungi, algae, and higher plants are eukaryotic organisms. The first drawback of this classification is the grouping of prokaryotes and eukaryotes in the same kingdom. The second drawback would be fungi is heterotrophic. Plants and algae are autotrophic, while protozoa and bacteria are unicellular. But vertebrates and invertebrates are multicellular. You can see all of them are mashed up into groups, where unicellular and multicellular organisms are packed inside one kingdom, and while autotrophic and heterotrophic organisms are grouped inside one kingdom. So that is another drawback of this classification. Hence arose the third classification system. The third classification system is known as the Ernest Heckel classification, also known as the Three Kingdom classification. Heckel saw other organisms like fungi, protozoa, bacteria and slime molds were in together with Animalia and Plantae in the previous classification. So he pulled them off and put them into a third new group known as Protista. Whereas he put the bacteria, slime molds and protozoa into the Protista group. This was how the third group known as Protista arose. This classification also had a certain drawback. If you consider bacteria is a prokaryote, but a slime mold and protozoa are eukaryotes. So, having them in one group is like a mashup. Due to that problem in the previous classification, hence arose the fourth classification known as the Copeland classification. The Copeland classification, also known as the Four Kingdom classification. Copeland saw the limitations of the Protista kingdom. He pulled off all prokaryotes and then put them into a group called as Kingdom Monera. All prokaryotes, eubacteria and archaebacteria in the Monera kingdom. So this is the fourth kingdom formed, and this comes under the Copeland classification. And now we have arrived to the most important classification of all, so I will take some time to explain this in detail. This classification is known as the Whittaker classification, formed by Whittaker, also known as the Five Kingdom classification. Whittaker observed all organisms. They can be prokaryotes or eukaryotes. Prokaryotes are unicellular. Eukaryotes are both uni and multicellular. Now, unicellular organisms have got one kingdom. Prokaryotes have got another separate kingdom. Prokaryotes got Monera and eukaryotic unicellular organisms got Protista. Eukaryotic multicellular organisms have got kingdoms, plants and animals. But plants can be both autotrophs and heterotrophs. Autotrophs have got a kingdom known as Plantae, while heterotrophs have got a kingdom called as Fungi. R.H. Whittaker observed evolutionary relationships, studied all theories, and then he grouped organisms into these 5 kingdoms: Monera, Protista, Animalia, Plantae, and Fungi. So, what are the criteria Whittaker considered before classifying them? Let us see. These were the classifying criteria: the cell structure, dividing them as prokaryotes or eukaryotes; the thallus organization, whether they have a thallus-like body or an organ-based body; their nutrition, autotrophic nutrition or heterotrophic nutrition; and their forms of reproduction; and finally, phylogenetic relationship. When you say phylogeny, it means the evolutionary relationships. So let us see the Whittaker classification's characteristics and how it describes the organisms. If you consider this following table, it has got characters and all 5 kingdoms. If we consider the characters one by one, we'll first go with cell type. Monera has got a prokaryotic cell. Prokaryotic cell type means the cell is in the pre-evolutionary state, in which the nuclear membrane is absent. While others, the Protista, Fungi, Plantae and Animalia, all have eukaryotic cellular organizations. Next, if you consider the cell walls. In Monera, cell wall is present, but the building unit is not cellulose, but instead peptidoglycan is present. And if you consider Protista, the cell wall is present in some organisms and in some it's not. For in diatoms and dinoflagellae, there are cell walls. But if you consider a humanoid, the cell wall is absent. Also in fungi, the cell wall is present, but the basic building unit is not cellulose, but a special thing called as chitin. If you consider plants, they have a cell wall made of cellulose. And if you consider Animalia, they do not have a cell wall. As I said before, for Monera, it is a prokaryotic cell. Hence, it is in the pre-evolutionary state and the nuclear membrane will not be present. Hence, the other evolved organisms will have a nuclear membrane. Next, if you consider the body organization, Monera is a unicellular organism, while Protista too. Fungi in the sense is a multicellular organism composed of loose tissues. Plantae has tissue and organs, while Animalia has tissue, organs and organ system. This particular example of body organization can explain the evolutionary pathway to you easily. Next, let us consider the forms of nutrition. Look at the nutrition of Monera and Protista. Both have dual nutrition, autotrophic and heterotrophic. Autotrophic in the sense, it can be two, chemoautotrophism or photoautotrophism. If you consider Monera, Monera has both chemoautotrophism and photoautotrophism. Chemoautotrophism is the synthesis of food using chemical energy. Meanwhile, photoautotrophism means synthesis of food with the help of sunlight. And an important point, Protista does not carry on chemoautotrophism. It only carries on photoautotrophism. In the meantime, fungi and Animalia are heterotrophs. They depend on other organisms for food, while Plantae is an autotroph. It can produce its own food through photosynthesis. And the final form of classification is Carl Woose's Sixth Kingdom classification, also known as the Carl Woose classification. The specialty of this classification is that Carl Woose did not touch any eukaryotes. Instead, he only worked with prokaryotes. He divided the kingdom Monera as eubacteria and archaebacteria. In this classification, eubacteria, also known as the true bacteria, are the bacteria whose cell wall is made of peptidoglycan. In the meantime, archaebacteria, their cell wall is not made of peptidoglycan. There is one more difference. Archaebacteria is mostly made out of lipids and proteins, while eubacteria has lipids found in very simple chains. Therefore, in archaebacteria, lipid blanching occurs, and due to this, they are found in very harsh habitats, for example volcanoes or hot springs. In Woose's classification, organisms are further judged and by their 16S rRNA. Oh my god, what is that? So, if you consider rRNAs, they are known as ribosomal RNAs. We have already studied about 70S or 80S ribosomal RNAs. But as you divide a compound and divide it and you go into the smallest unit, the atom. Same as that, if you divide a 70S or 80S ribosomal RNA, the final or not the final, a special kind of subunit you get is known as the 16S rRNA. And you already know 70S rRNA is in prokaryotes and 80S is in eukaryotes. Woose saw all eukaryotes, archaebacteria and bacteria had 3 different types of 16S rRNA. So he put all of them, the 6 kingdoms, into 3 major domains. To beautifully represent this, I can draw this as a tree with 3 branches. And as we know, the domain is a higher taxonomic category, and he gave 3 names for the 3 major domains of life known as Eukarya, Archaea and Bacteria. And also, Woose's classification reveals us one truth: the root of evolution, or also known as the common ancestor. Now we all know for a tree to grow, its root is a main important base. So, our common ancestor of all evolution is same as the root of a tree. So for all these 3 major kingdoms, the common ancestor is a progenote. So if you see, these progenotes are our common ancestor of these 3 basic branches of life. The domain Eukarya, Archaea and Bacteria. I hope you understood this, and let us meet again in another video. Until such time, it's me Brainiac signing off. Thank you for watching and bye.